Oh. Hi, everybody. You're at Jimby Live. I'm Rachel Horak. I'm staff in the ASM Education Department, working on programs for faculty to learn how to teach a little bit better. So we're going to talk today about Jimby Live's episode. We'll be teaching with TED, a curated set of TED Talks and discussion prompts for microbiology and cellular biology courses. Really excited for this conversation today. Our speakers today are Diane and Eileen from Kansas University, and moderating the Q&A will be Editor-in-Chief of Jimby, Stanley Malloy. Hang on a second. Okay, so this is the title of our presentation today and showing you all the people that you're going to meet today during Jimby Live. So Jimby is the Journal of Microbiology and Biology Education. It's a completely open access journal from the American Society for Microbiology, and that means that you can read any of the articles without any fees. Okay. So it's a great journal. They just published a recent issue within the last week or two, and I am so excited to dig into that latest issue. I think you should be too. One exciting program that ASM is offering this year for educators is the ASM Conference for Undergraduate Educators. You can scan the code to learn more. It takes you to the homepage of our website. And one exciting update is that we're going to not only offer one conference this year, we're going to offer two conferences. So we're going to have a summer series called the ASMQ Summer Series in July and August, completely online through Zoom. It'll be very easy to use. And I'm really excited. We're going to announce our plenary speakers next week for this conference. Also, we're going to offer our signature program of the in-person meeting November 21 to 23 in San Antonio, Texas. So we're currently accepting proposals for talks and sessions and microbrews at this time, and we would love to see your scholarship at one of our meetings. Okay, with that, I'm going to turn it over to our speakers. And when you have questions for our speakers today, you're going to put your questions into the Q&A box, and we'll address all of them through a moderated Q&A at the end. Take it over, Eileen and Diane.
All right. Okay, I'm just going to share my slides here. All right. Does that show up all right on your end?
That looks great.
All right. Okay. So hi, I'm Diane.
And I'm Eileen.
And we are coming to you live from Lawrence, Kansas, home of the University of Kansas. So thank you all for joining us today, especially this time of year. I don't know about you, Eileen, but I'm feeling it.
Oh, for sure, for sure.
Okay, so we are happy to talk to you today about teaching with TED or teaching with TED Talks. So we wanted to start with just a show of hands. How many of you have encountered challenges in engaging your students in course content, getting them excited about their course content?
I think you can push your reactions on your screen.
Yeah.
There we got
Okay.
a few.
seeing some reactions here. Okay, so the TED Talks that I'm going to talk about today, I have shared in Biology 416, my cell structure and function class. And while I get super excited about cell biology, my students don't always get as excited as I do. So I've been thinking about ways to get them more excited, to kind of hype the course content. And so what I turned to, and what Eileen has also turned to, are TED Talks. So I didn't even realize TED was an acronym, to be honest, at first. But
it was a guy.
Yeah, I don't know. So it turns out TED stood for originally technology, education, and design. Now the topics are much more broad, but that's how these TED Talks started. So they're appealing to me as an instructor because they're not too long. So they're on average about 15 minutes. They're suitable for a broad audience. They're on a variety of topics. However, when I was starting to do this, the daunting part about this was that when I searched biology TED Talks, 877 talks are available, at least as of like last week. So that was a lot. 66 of those come up if you're searching for microbiology, 242 if you're searching cell biology. So that is quite a lot of options, and that can be rather daunting. And so Eileen and I have whittled down these 66 and 242 to try to find some really some talks that we thought were really appealing for these different class audiences. We wanted to acknowledge we are not the first ones and certainly not the last or only instructors that are using TED Talks in our courses. And so we wanted to give a shout out to Fergasso and Capozzi, who have used TED Talks in their courses, specifically focused on ethics.
Mm-hmm.
And I think that's a feature of these TED Talks, is that they don't have to all be completely focused on the science. They can also bring in some other topics that are important for our students to think about.
Yeah, and we'll kind of highlight that with ours.
Absolutely.
Yeah.
Okay, so we turn to TED Talks, but then, okay, what do we actually do with these TED Talks? So what Eileen and I both have done probably the most is use these for online class discussion boards. We find that these online discussion boards allow us to promote a deeper understanding of the course content. They build community. So our students are working in groups. They are grouped online into smaller subsets of the course, so they can talk about the material in these smaller settings. We find that it engages their critical thinking skills, so they really have to think more deeply about what they're learning. They practice their communication skills, so they actually have to write something about what they've read and what they've learned. We find that these discussion boards are provide better access. They can be very flexible, and we think they've been a safe place for diverse perspectives, in part because it is this smaller group from a larger class, and those are stable over the course of the semester. And so overall, we found that these online discussion boards are a great platform for taking classroom content and making it more applicable to our students' lives.
And something to kind of build on what you're saying there, Diane, is both Diane and I teach very, I would say, large or jumbo size classes. I mean, I don't know what the range of your classes in the hundreds.
Yeah.
And then some of the classes I teach bordering 350 to sometime, well, I think the largest one was about 600, but that's been kind of whittled down to in the 300 to 400 students. So yeah, this online discussion format allows more students to engage with the material in a smaller group, allows you to have some of those quieter students that aren't going to participate in class. They're participating in these groups. And like you were saying about building community, we have these students built into like 8 to 10 person groups. And sometimes those groups, we pull them into the classroom and do things like group quizzes or group work, and they stay within that group. So they're getting kind of an online and in-person interaction with those groups. So yeah, it's kind of a nice format.
Okay. How many of you have done discussion boards before in your classes, have run these online in your LMS?
Yeah, maybe just, yeah.
Any of you show of hands? All
Yeah,
right.
a few thumbs
Okay.
ups, good.
So I think especially as all of us had navigated teaching remote online because of COVID-19, probably a lot of us have experience now with discussion boards. But we just wanted to mention that creating these discussion boards in Canvas or Blackboard, whatever your LMS is at your institution, can be pretty easy, pretty straightforward. So you can go in there and either assign groups of students that you want to be in groups together, or you can just say, do this randomly. I want this number of students in a group or this number of groups in the class. You can grade them or not. I probably would recommend grading them. I think that is going to get much better participation. There are different options for moderating the content. I think both Eileen and I have tended to have our graduate teaching assistants grade these, but you could certainly moderate them yourselves. And I think we found that having really clear instructions up ahead of time and consistent deadlines has led to success with this format. So giving them a rubric about how you're going to grade the discussion posts and the replies, and setting deadlines so that you do, so students know what to expect each week and so that they do participate early and often.
Yeah, I think just what you said, Diane, having those deadlines, usually what we do is, and I think we both do this very similarly, is divide the score into kind of two components: an initial reply by the student, so they're filling out the prompts, and then they have a part of the score is them replying to someone in the group. So setting that first deadline for that reply gives all the students in the group the access to having somebody to reply to. So that can sometimes be an issue. Yeah.
All right. So I wanted to go over and dive into now the actual TED Talks that I selected for cell biology. So I teach this cell structure and function class mainly in the summer semesters. So that's an 8-week course. We use the Essential Cell Biology textbook, and I feel like these TED Talks align well with the layout of those chapters, but I think it could be used in any kind of mid-level cell biology course. My course has the 2-semester intro bio sequence as a prereq and also genetics. So we have sophomores through seniors in this course. So we start off talking, we watch Geraldine Hamilton's TED Talk about body parts on a chip, and students find this really fascinating because we've just introduced the idea of model systems here. And they all kind of love the idea of having, well, many of them love, some of them are a little creeped out by the idea of having, themselves on a chip. But I think it gets, especially the pre-health students, real excited about the future of medicine and precision medicine. Then we move in and we hear from Elizabeth Blackburn about the study of telomerase and the discovery of telomerase. And I think one thing that's really great about her TED Talk is that she not only talks about the science, but then she also gets into talking about the effects of stress on telomeres and gets into discussions of mental health. And that's been led to really fruitful conversations on the discussion boards where students talk about how they might make changes in their own lives as a result. We look at David Liu's TED Talk about base editing and possibilities there. And we'll see an example of the prompts for his TED Talk in a minute here. And then we dive into Alejandro Sanchez Alvarado's TED Talk, where he discusses approaches to research. So he talks about some of the work that he's doing exploring marine biology. But I think what students love about his TED Talk is that he talks about kind of how many times throughout his career he feels it's common that he feels stupid or that he doesn't know what's going on. I think that's relatable to all of us. And the students really love that and they are surprised by it. And then it just makes it so relatable to them, and it normalizes that feeling for them, which I love. And then the last couple TED Talks we discuss are focused on cancer biology. So we talk about, we see Carl June's TED Talk where we learn about CAR T cells and their potential in the clinic, and Joshua Smith's new technology he's developing to detect cancer earlier. So we kind of leave them with this very hopeful, uplifting stories. And again, connecting to the topics in our course. Show you here some examples of the prompts. This is from David Liu's genome editing TED Talk. And I just wanted to highlight here that with our first question here, we're focused more on the science. So we ask students to sort of write a paragraph or two about this topic if they were going in to update their textbook. So this topic was not in the textbook. So what would they put in there about it, and making sure they understand the science behind it. But then we also get into some questions about sort of the funding of the research or the setting of where the research takes place. some of the ethics, some of the concerns about where this research might go in the future. So we try to touch a little bit more on the biology, but also on the social aspects of it.
Yeah, that's cool. And one thing to point out in the paper, we have in the supplemental figures, all of our prompts from the ones that we've given in the classes. So
Exactly.
yeah.
So hopefully that makes things easier for everyone.
Yeah. So in the microbiology class I teach it all 3 semesters, 1 semester in the fall. It's a pretty big class, usually in the upper 300s. Summertime, it's online, just like Diane's, all online. And then in the spring semester, it's a flipped classroom. So I use the TED Talks with an online format in some, but in some we do in person. So for the fall big class and the summer online class, it's an online assignment for the students. So I think it's in the neighborhood of 5 to 10% of their total score. In the spring semester, it's nice because we can bring them the TED Talks into the classroom. And then it's fun because I get to actually see their reactions to watching them. So that's kind of nice. But we do in-class discussions. So we use the same prompts, and they turn it in as a group in that flipped classroom format. So I don't do all of these, but I've used them all at separate times. I typically have about 4 discussion boards in a semester, regardless of the format. And then again, these are linking some of those content-related material to what we're discussing in the classroom to kind of how it applies in real life and more meaningful for the students. So one of the ones I use is Bonnie Bassler's talk, How Bacteria Talk. And she actually has 2, she has like a follow-up, more mini TED Talk that I show as well. So it's kind of an update on the information. So that's talking about bacterial quorum sensing. And it's a great, really, she does a fantastic job of bringing this to a layman's audience and gets the students excited about that idea of quorum sensing, which you can bring into a number of different ways in the microbiology class. But one thing she highlights is how looking at prokaryotes, how they communicate, helps us understand eukaryotic cell signaling. So it kind of also brings in that evolutionary idea to the classroom. There's 2 I kind of oscillate between in the semesters when we're talking about antibiotic resistance. Both of them kind of highlight, the pretty big social issue of antibiotic resistance and how we're overusing them. That's Marilyn McKenna and Ravina Kular. Both of these are great talks highlighting the misuse and how we need to be better stewards of our antibiotics. And what I like about this talk is you have a lot of pre-meds, pre-pharmacy, pre-nursing, pre-health. A lot of these students are going to end up in the healthcare field, so you feel like you're weaponizing them with good information, and so they'll be good stewards of the antibiotics. But also just in general, students realize that I don't need to run and ask for antibiotics for my cold. So they are, and then also a lot of them come back and say, hey, I don't buy meat. I check where my meat's coming from. I make sure it's not using too much antibiotics. Another one I'm going to highlight in a minute is Stephanie Strathy's TED Talk. So she and her husband were traveling in, I can't remember where they were traveling, but I think over in the Middle East. And he got a antimicrobial bacterial infection with Acinetobacter baumannii. And so it's kind of her story, which is pretty harrowing, where she found phage researchers who would send her phage and the bacteriophage to fight her husband's infection. And so it's just a fantastic story. It makes me cry every time I see it. I've seen it like 5 times, maybe 10 times, and I always still get very teared up when he comes out at the end. Like I'm surprised he's still alive. I don't know. But it's a really great talk that not only brings in bacteriophage, but also antibiotic resistance. And what's really cool about Stephanie is she has told us, or she talked to my class once about being neurodivergent. So it kind of shows the diversity in science, but also how her husband was the first phage, like clinical trial of IV phage in the U.S. And now she's started this whole institute on phage. So it's really quite a massive story. And then Rob Knight's talk, I think it's from 2017 or 2015, kind of the beginning of the microbiome revolution, talking about the students get to see, understand how microbes are all over them and why they're there. So that one's a really great one to introduce with the microbiome, or even if you're just introducing the class in general, just getting students excited about microbes and bacteria. And then during the COVID pandemic particularly, but even before that, I did one on vaccines and kind of vaccine hesitancy. These are two really good ones by Hardy Larson and Lindsay Diamond. They do a very nice job navigating this sometimes politically charged topic, but they do a really nice job with that. And so, and now with like measles outbreaks coming, we talk a lot about that in the classroom.
Yeah.
So this was a really good one because it talks about why people don't are hesitant about vaccines, talks about herd immunity, kind of all the whole gamut around vaccines and public health. And then this next one by Nicole Cabrera Salazar, The Future of STEM Depends on Diversity, is one I use in an identity in STEM prompt. And that one I'm going to talk a little bit more about. So this STEM identity talk is really excellent. She's a young researcher, and she talks about her experiences getting into STEM, coming from a minoritized community. And so the way I've used this in the classes, I'm fortunate to teach the intro bio class that Diane referred to. So the first semester freshmen, we offer this as kind of an extra credit for an exam, like a reflection. So we ask them to describe how they relate to the field of science and their experiences that have attracted them to the STEM field. So we want them, freshmen are losing all their identities that they've had all their life of a football player and as a, drama kid. Now they're having to re-identify themselves. So also kind of asking them to reflect on this new building of their identity. So we have them reflect on that, discuss how they're going to see themselves in the field of science and who influenced them. Spoiler alert, a lot of times it's their high school teacher, which is great. And then we ask them to tell us about their long-term goals. What are you trying to get into? And so I do this in that class, but then I reintroduce it as a TED Talk in my Bio 400 class. Oftentimes I'm seeing these students again in the intro micro class, which is the Bio 400. I've got sophomores, juniors, and seniors. So I'm asking them to revisit this question to kind of, signify the importance of reassessing your identity, but also as a way for me to kind of get to know them a little bit better. That class is a little bit smaller, 300 to 400 students. So I asked them to do it again. I think this one might actually be a bonus point assignment because I read through them. And I find that this is an opportunity for me to become aware of their experiences and then also see what their goals are. So for example, although it's a little blurry here on the left, this was a student who came back in microbiology, but I knew him from the intro class and I knew he was interested in biotech. And so I read his prompt and I was like, he's still interested in that. So I sent him a note and said, hey, come see me and let's talk about ways to get into research. And we found some opportunities for him here at KU. So he's super excited to start those. So it's those little touching points that you can go back to the students and learn about what they're doing. For the Bio 150 students, those intro freshmen, I also model the response because I want to demonstrate what they should be doing to answer these questions. And so I answer them from my perspective. And I think that's an opportunity for them to see my identity in science and how, right? Sometimes I feel stupid and I had imposter syndrome and I loved my high school science teacher and my mom was a nurse. So you start to humanize yourself in science. And I think the students really appreciate it. And funnily enough, like I was talking about my high school science teacher, Mr. Aronson, and a girl came up to me this semester and her sister went to the same high school and knew who I was talking about. So that was kind of fun too.
He's a longtime teacher.
Yeah.
Yeah.
He retired this year.
That's probably good.
Yeah.
Let him enjoy that. Well, yeah, and it's funny because, you said the students enjoy this, and I can vouch for that because this semester, some of your former students were actually talking about this
Yeah.
PM activity and how much they enjoyed it and how meaningful it was to them.
Well, I don't think anybody, when I was going through my undergrad experience, said, think about your identity in STEM. And they never, you didn't really think about it till you were in grad school and then whatever. But I think if students kind of intentionally process that, it's not a bad thing.
Yeah.
Another way I've used the TED Talks, and this was a TED Talk by Irene Eskla, the Antibiotic Resistance Basic Science to the Rescue. I use it preparing students in the spring semester for their oral presentations that they're going to give at the end of the semester. And so the way I do that is trying to get them to understand how science papers are written for a scientific audience, but when you're giving science communications, that's not always the case. Sometimes you're trying to convince somebody to fund you, or you want to just explain to your mom what you're doing in your lab.
Right.
And so this kind of shows that different types of science communication. And so in the class, what we're asking them to do is take information from these papers, distill it down to an audience that you can, like a layman's audience for your presentation. So I think that this TED Talk does a really nice job of explaining a complicated research article. So I have the students read the research article at first, and they kind of struggle with it a little bit. We go through the figures and the results, and then we watch the TED Talk, and then they can see, oh, this is how I can distill that information down. So that's kind of a really fun way to bring in this TED Talk for some science communication skills building. And then some of the unanticipated positive outcomes of bringing the TED Talks into the classroom. So I have a Twitter account or X or whatever, and an Instagram account for my class. And I will tweet out articles, and Stephanie Strathy's talk and kind of her promotion of phage research and stuff it caught my attention. And so I would tweet stuff out. And then pretty soon I noticed that I followed her on Twitter, and then I saw that she was following my Twitter account, and I was like totally geeked out about it. And so I DM'd her and I said, hey, I use your TED Talks in my class all the time. It always makes me cry. And you can see here's our little message back and forth. And apparently she keeps crying about it too when they watch it. But she offered that she would do a Zoom Q&A for my class. And I was literally walking to class doing this DMing back and forth with Stephanie Strathy. And I walk into my class, I'm like, we're going to have Stephanie Strathy come talk to us. So she was very, very gracious and did a Zoom call with my students, and it was just absolutely so much fun. I was kind of panicked the students weren't going to give questions or be brave enough to ask her, and so I was asked them to kind of send me their questions just in case, and I had a bunch ready to go, and I didn't even get to ask her a single question because they were just raising their hands, and we actually went a little over time. So she's been super gracious. I actually just recently bought a few more copies of her book, and she's gracious enough to autograph them, and we give them out to the top learner in the class, and the students are usually pretty excited about that. So that was kind of I'm not saying Stephanie will do don't tell her I said she would do a Zoom with me, but she's been fantastic partner, and it was kind of a fun thing.
Yeah, what a cool connection. All right, so we do all of these things. What do the students think about them? We have found, we have different types of feedback here. So on the left, we have the anonymous feedback that students have provided through our teaching evaluations each semester. And I think from what I can tell, the TED Talks might be the favorite part of the class.
Maybe.
So they tell us, that these helped them in their learning. It helped them, understand things, but also it helped motivate them. It got them more excited and made them continue studying, even if they didn't find learning about transcription that engaging. We also both heard from students over the years that they were using these TED Talks to then go and talk with their families and their friends. So one of our quotes here is one of our students went and talked with their husband each week about the TED Talks. And So that to me is like my favorite
Yeah, I
part
would
of
agree.
this, is
I would
that
agree.
it's helping those students go into their communities and share the science.
Yeah, that's fantastic.
And then some of the students that spoke to us on the record that, you know, again, mentioned some of these same things they liked about the TED Talks, and also said they wished more of their upper level courses would incorporate this type of thing. So
Yeah, I
that
think
was great.
we sometimes forget how the content in the class is applicable. Like we
Mhm.
know how that is, but they
Right.
don't, and we forget they don't understand that. So
they need
bringing
to
it
know.
from the textbook to this is how it's being done in the real world makes it really exciting for them.
And so now we're ready to take questions. I did, though, want to mention, so if you're thinking about doing this, baby step it.
Yeah.
So start off, you know, add one TED Talk in, one discussion board, or, you know, use one of our sets and keep it simple. So Eileen has been doing this a bit longer than I have, and so she has now developed this really cool set of kind of built out assignments that go above and beyond, but I haven't done it as long, so I'm still sticking just with these discussion boards. But over time, you'll kind of start to see cool new avenues to take these assignments down.
Yeah, I would echo exactly what you're saying. Don't feel overwhelmed. Maybe one at a time, find something.
Mhm.
I think mine, it was like antibiotic resistance is kind of where I started because and just, oh, this would be neat to share with them. And I might have just started sharing them before I even built it into an assignment.
Yeah.
So yeah, take your time with it. Yeah.
Well,
Right.
thank you very much, Eileen and Diane. That's really very inspiring, right? And so I have a couple questions before we start, and then we'll ask questions from the audience, right?
Sure.
But one of the things I wanted to emphasize was a comment, and that is, you know, the Stephanie Strathy book, in addition to the heart throb of knowing that your husband might die, right?
Right.
It really emphasized how the international scientific community came together to solve this problem. And that's something that we don't tell students about very often. Did they appreciate that?
I think they appreciated that, and I think that what they also really kind of hooked onto was, especially in the book, the students that I've heard back from that read the book was that all that emotional stuff that she was going through in the hospital. You know, I think it really connects students to her husband was in the hospital with ICU. I mean, for like 9 months. He was in septic shock like 6 times or something ridiculous. I can't believe he's, you know, he's still alive. But I think they connected to that. But just for them, it, you know, I again still tear up. I teared up this year when I showed it, but you could just see that kind of the emotional connection hit them really hard with that. But yeah, I think that part about you had the Army, you had Rye Young down at Texas A&M, you had phage researchers from all over. They get really excited about that whole idea that phage are everywhere.
Yeah, that's really good. As a sideline, I know Stephanie and Tom, and I
Yeah.
knew them before all of this happened. So
Yeah.
it's
I wondered when
especially-
you said you were up at up at St.
Yeah.
Yeah, because she started that whole iPath or and she and Chip have kind of dove into that. I think that's amazing that she started with I just want to save my husband, and now the clinical trials and everything they're doing is just fantastic.
Right, right. One of the other stories that you mentioned actually does this really nicely as well, and that's Rob Knight's story, right? Because that's a case where he develops tools and gives them to people around the world, right? So I think that's also just another kind of inspiration for the way science should be. Right?
Yeah, and you know, you're actually tickling my brain a little bit about adding that to some of my prompts, you know, talking about kind of the internationalization and the globalness of science. Doesn't have bounds or politics attached to it. Yeah.
Do either of you have any data on how the TED Talks going through this process influences student success in other course content? So, I mean, in addition to getting them engaged, does that engagement spill over into the other things that they're learning?
Well, something that Diane and I realized, she talked about that TED Talk she does in her class about CAR T therapy. When in the micro class, we have, you know, maybe 4 chapters on immunology, and I briefly talk about CAR T therapy, and they get really excited about it. So I love that it's scaffolded into your classes as well. So, but I don't know that we have any specific data. That would be interesting.
Yeah, I don't think I have any, but that's an interesting idea.
I mean, I think you would have to have a control group to answer that properly. Which
Right.
means that some kids would miss out on this great experience. But,
Yeah.
but I do think it fits very nicely with a tremendous amount of education literature on the impact of relevance in educational processes, right?
Yeah.
Um, you have a set of some really nice talks. Have you thought about updating them? Do you regularly follow the new TED things that come out to see if there's something else so that it might keep it as current as possible?
Yeah, so I try to keep an eye on those. The other thing that I do is that, some of the talks are a little bit older, but they're still really good. So what I do is during the weeks when the students are watching those, I will send them like, okay, here's the TED Talk, and it was from, 2019 or 2022. But this same researcher just published this new paper, and here's a link to it, or here's the PDF. You can go see now the update, or I'll summarize the new steps in the field, and the students get excited about that. And sometimes when I haven't done that, they've even,
They'll ask
Googled
for it. Yeah, they'll ask for it.
And they'll come to me and say, "Oh, I found this cool, and do you know that now they've taken it this step further?" So I love that it's getting them intellectually curious.
I've had them send me TED Talks. They're like, have you seen this one? I'm like, yeah, I saw that one. So that's, it does, we do see that kind of feedback from the student.
Yeah.
Yeah, that's really nice. I mean, I think sometimes 2022 doesn't sound very long ago for most instructors, but for students, it may seem a lifetime ago.
Yeah, they're like, this is old, 3 years ago.
You're like, science moves a little slow too, so there might not be that much progress.
It's from the 1990s.
So this next question ties in some ways to your comments about Stephanie's book, and that is, I wonder if by listening to these talks, it gets students to read more scientific literature on their own. You actually mentioned that a second ago, and to follow up on the talks, but also, read some of these popular books that relate to the talks. I mean, Stephanie Strathy's book's one. Ed Yong has a number of books.
I was just going to say that one. I've talked to students. Yeah, I've contained multitudes. After the microbiome talk, I always am promoting that book for sure. Yeah.
Yeah. Do students actually go read these books?
I've had several students who have read Ed Yong's book for sure. I have one student this semester. He's like, I'm really trying for that Phage book. I'm like, you can buy it on Amazon. If you don't get it,
library
you can borrow my copy. It has some. So yeah, there's a lot of communication with students who have read some of those books. One time, I always promote and say, if you share scientific literature in your responses back and forth, then you could get extra credit, so sometimes students will dig a little deeper and do that. My class, especially the intro to micro, you have a pretty big range from sophomores to seniors, so their ability to go out and grab that resource is a little bit limited, I think, or they just lack that experience. But it might be an interesting way to kind of teach them to start getting into the PubMeds and doing those literature researches. But definitely the side of it where it's more accessible, like, although I haven't done one on it, there's like a PBS series called The Poison Squad. It's over kind of food microbiology. And so that I've recommended that to students. And I hear that maybe Stephanie's got something coming out on PBS or something. We'll see.
Little bird might have mentioned something.
She's been a gold mine for me, that's for sure. Her stuff is like, it's always easy to tweet out her stuff. It's been great.
That's wonderful. So we're going to shift over and have some questions from the audience, right?
Okay.
And the first question is, based on the topics covered using the TED video, did you have any assessments related to those specific topics? And can you share those assessments with us?
So I think for me, the topics, the assessment was the discussion boards. themselves, because those were graded and they were graded based on a variety of different parameters. But I don't think I really had any, like on the exams or the quizzes, I don't think I had any questions about the TED Talk specifically, because they were so much kind of beyond what was in the textbook. So I don't think I can help in that area so much.
I would say in ours, it was a little bit of both, because, you can't understand Stephanie's talk on phage therapy if you don't know what a phage is and don't know what a lysogenic and lytic cycle. So certainly that part is on the exam, like I give them background on that, and then we watch the TED Talk, or, the microbiome, understanding that. So there are some concepts that they have to understand, and so they'll be assessed on that. I haven't linked or looked at, did that help or not? I definitely think it makes them more interested, and we all know if a kid is interested, then it's going to impact their outcomes for sure.
So the next question ties to this as well. And this is from a professor at a college in Florida. And they're teaching a new microbiology class. And they're really interested in using creative assignments to help students apply real world applications of microbiology. So the question is, can you suggest some ways that you can integrate the TED videos into the course to enhance the student engagement and understanding? The presumption here is this means in addition to what you're doing already.
I could think that, and it's kind of brewed from this discussion just now, like make the students bring the TED Talk to you. Say, okay, for, whatever, tell me why you think this is relevant, explain how this links a content from the class. So you could have some sort of like, have them, Ed Young has like a playlist of microbiology, like kind of relevant videos that at the end of the semester, just again, like a bonus point for an exam, I have them read those or pick whatever topic they want, watch the video, convince me that I should watch it in a written assignment, and say how, because in my mind, we don't have enough time to talk about everything in the semester about microbiology. So I'm asking them to do that. So I could see that being a useful assignment, because take what you've learned, find something related and like it.
And I assume for cell biology, that's also true. Like you said, a lot of the things that are covered here, like CAR T therapy, aren't in the textbook, right?
Right. I could also see asking them to bring in, other news media. So not even just TED Talks, but saying, so something we do, I teach immunology also. And so one thing we do at the end of the semester is we ask them to address immunology misconceptions. They create shareable media that uses their newfound immunology knowledge and addresses those misconceptions. So I think getting them to apply things to their everyday lives is crucial in getting them excited about the topic.
Yeah, for sure.
This ties back to a comment that both of you made, and that is the concept of helping students communicate science better to a lay audience, right?
Yeah.
And I wonder if you have any feedback on how effective that has been.
Well, we, like I said, in the spring semester, we do a presentation. So it's still a big class, but, and you're coming next week to help with it.
Yes, I am.
So remember, don't forget.
Yes.
So we have a nice classroom set up. So we have 20 tables that each have their own monitors. So the end of the semester, the students have been working on a group presentation on anything they want to talk about that's microbiology related. So we just did some of them on Tuesday. We had students talking about raw milk versus pasteurized, kind of a relevant topic. And that was really fun, because the students actually asked if they could go in the lab, and they inoculated plates from pasteurized and raw, and that's part of their presentation. So I think they all did great. They scored well. We have faculty come in and evaluate them using a rubric, but I think it helps them see, like, this is how TED does it, and this is how I can explain it too. And we really highlight that, we want to make science more accessible, and we do that by taking away the language that's kind of a barrier for people to understand it. So they present it in a way, we say that you know, even though Diane has her PhD, pretend she doesn't know anything about it or is just a basic understanding. So it does help them with that.
Yeah, I think in cell biology, we see, you know, we saw some of that feedback that the students are taking the TED Talks and using those to communicate out to their families and their friends. And then in immunology, I see that absolutely. I think there's so many topics in that course that are really relevant. So we've been talking a lot. So Kansas right now has quite a few outbreaks, especially in the southwest, western parts of the state of measles. So we've been talking a lot about vaccination and how vaccines are developed anyway. So I think it is effective because the students, they come back and they tell us stories about, you know, who they've talked to and what they've said and how it's gone. And they don't always change people's minds, but they make an attempt at it.
Yeah. I mean, I think that's great because for a lot of people, this may be their only contact with an academic perspective, but it's got to be in a way that they can understand. So I applaud you for helping people do that. And back to the TED Talks, Bonnie Bassler is probably the world champion at doing that. So
Absolutely. She does a great job. And the nice thing is she actually had that follow-up video. So that is fun to give the students that information. So yeah, that's great.
Right. So one of the things about this is it takes a lot of effort to do something like this because you really care about student outcomes. And so we have a question from the audience. How much time per week do you spend monitoring and responding to discussion boards?
So I think it depends. So again, we have pretty big classes, so we have a really decent amount of GTA support. I will say that STEM identity one, I read them all myself, just not in the big class, but last fall, I mean, and this semester I had 180 and so 300 something. So, I mean, you get pretty good at breezing through and trying to find the important, impactful points. What's nice for us having that GTA support is that we can divide the class digitally into sections. So my TAs this fall had the same group of students, but it was also kind of forcing them to build that community. We always tell them, give us a couple weeks to grade them. So they have they're open. The way I have it set up, it's open until the first exam, and then that's when kind of the due dates show up. But then we tell them just give us a little bit because it does take a little bit to dig through those. And it also depends on, I'm really skimming and looking for some key points. I'm not grading them on their grammar. I do say, you know, there is part of the rubric is grammar because until I did that, they weren't capitalizing their sentences or I'm like, this isn't we're not
readable.
Texting. Yeah, this is professional. So, but, you know, it takes a little bit.
Yeah. So I have a graduate teaching assistant that helps me grade these. So I personally am not spending much time. I usually read some of the first ones just to get to know the students. And then if the GTA brings anything to my attention, I'll go in and read those either because it's really good or some concern about a student. But, you know, I'm guessing that they probably spend 2 or 3 hours a week reading through those and grading them.
Yeah. But we've gotten to a good point where we've taken some other things off of their load, like we use GradeScope for grading, so we recognize that it's taking time, and we ask them, and we kind of highlight with them, especially some controversial ones. We help guide them on how to manage those. We ask them to put comments in so they know somebody's reading them and that sort of thing. Yeah.
Well, that sounds like a great experience for the GTAs as well.
Oh, for sure. Yeah.
Will help them be better teachers in the future.
Yes. Yeah.
Well, in your paper, you mentioned that you also had done this in smaller classes, for example, during the summer. Did you have GTAs do it then?
Yes. We got lucky. Yeah, we have a GTA in the summer, so that's a nice opportunity. But sometimes we have to jump in and help grade too.
Yeah.
Yeah.
Because I guess for us, a small class in the summer is 65 students. So I don't know. Size is relative, right? Depending on your institution.
Yeah.
But yeah, because we have those still somewhat larger sections, we do get GTA support in the summer.
So is your success doing this with your classes? Are other faculty thinking about, hey, I should do this as well? In other words, is it as contagious as you might hope it would be?
Did you do it? Because I did it. Or
I
how-
did, yeah.
Oh, really?
Yes, yes. So we are an example already of the contagion.
Yeah.
And we just presented about this paper to our fellow biologists on campus a week or two ago. And already some of them are trying to think through how they might do this. Some of them also, we talked about not just TED Talks. So we started talking about how iBiology has some
Yeah, great videos.
cool videos there. If you want to dig more into the science side of it, then more the lay audience approach. So yeah, we hope that others will do this as well.
Yeah.
So if you were going to follow up on this, your next paper, this paper was a tips and tools paper. If your next research paper is going to come out, what would you do? Or maybe you're already doing it.
I got 2 other papers coming, Stanley, so we'll see. But I would say maybe some of the points that you had, like what kind of assessments, can you link it to understanding? Can you link it to success in other classes? Maybe at the very least, maybe a survey of student feedback of how they strongly agree that this helped them understand the content more. So kind of maybe measure the students' interaction with the TED Talks.
Yeah, I agree with all those points. And I especially liked your question about the research literature and how are they taking that and are they reading the literature more because of it? That one got me thinking. We don't have any like project underway with this right now.
No, yeah,
because
might be something
but maybe maybe on the docket, we'll see.
That's great. Well, I guess I'll also say I really appreciate your comments about self-awareness and this idea of feeling stupid. I don't think you can actually approach the world from a scientific perspective if you don't feel stupid, because there's so much out there that you're not going to know.
Yeah,
but it reminds me,
growing.
Right. But it reminds me, I was always, I'd sit as a student, I'd sit in the audience and listen to scientists ask questions and say, how can they ask that question? It's so smart. And then I went off and I had a mentor who is one of the few genuine geniuses I've met in my life. I've met a lot of very, very smart people, and this person had no qualms asking a stupid question. So I said to myself, well, I guess if my mentor can ask stupid questions, I can ask stupid questions.
Yes.
And that's where you find out that, some of what you think is stupid is not so stupid after all. So I think that what you're doing is a great opportunity for students to gain that kind of self-awareness. And I applaud you for that.
I love that about that one talk, and just letting them know it's okay. I say in one of my classes, I'm gonna ask questions if anybody has questions, and somebody's gonna ask a question, and then there's the rest of you sitting there going, how did he think to ask that question? But I felt that way in my classes, like I don't even know what to ask a question about. So
Right.
Kind of admitting that, students felt relieved that it's okay that you need to spend time later tonight processing this content and then developing your question. Then some people are just good at it and they can process pretty quickly to develop those. But yeah, I felt the same way.
Absolutely.
Especially in grad school, those first, you have to go to those presentations. You're like, well, I understood the title. That was about it. Yeah.
Well, thank you very much. It was really great learning about your work, and I think the conversation was also really fun. So thanks very much. I'm going to bring Rachel back to give us some final comments.
All right. Thank you.
Thank you.
Cool. Thank you so much. Wow, what an exciting tool that you could use in your classroom. And one of the things that I really like about it is they're freely available. Anyone, even if you're at a institution that may not have much money available for your students, you can go to TED Talks and do some learning there. And it's also, the other thing that I like about it is lifelong learning, right? You get the students hooked and they're going to learn some content that's new, and then they can go back to the website later and hopefully get them hooked on lifelong learning after they leave your classroom. So let's see, that concludes our conversation for GymB Live today. Don't forget, this summer, we will not have any episodes of GymB Live, but what you can do is come to ASMQ Summer Series completely online summer conference. We're going to have 6 sessions for between July and August. And you can go submit a proposal to give a talk and highlight your scholarship. And plenary speakers will be announced next week. Take a look at our GymB Live website for more episodes, probably coming to you this fall. And we'll send a follow-up email to all who registered with a link to this recording. So thanks for joining, everybody. Don't forget to go read GymB and continue learning. Thank you, speakers. We really appreciate your time with us today.